Okay, yeah, thank you for the kind introduction. It's really nice to be here and to present on this work. This is going to be less polished than Cole's amazing keynote yesterday, partly because it is very much a work in progress, but it's fun to share these results with you today. So the human reference genome doesn't need a lot of introduction. It is the cornerstone of everything that we really do in genomics as a community. Many of us can look back to its initial release as being essentially the point in time at which genomics started to grow up and become a real field. And it's important to us basically because it acts as a universal coordinate system, right? Everything that we do is in reference to that coordinate system. When we talk about genes, when we talk about annotations, when we map things, right? We use that reference as a lens to understand new samples, to understand differences. It cost originally, it's just interesting from a historical point of view, cost $3 billion, and it took an act of Congress to create. And it was, and I should mention, it was also an international project involving many countries. It was released in 2001, and it has been refined over 20 years. Initially, it represented just the euchromatin of the genome. It was about 2.4 gigabases of finished sequence. Today, it's much closer to a complete assembly, but it is still incomplete. There are about 200 megabases of sequence in GRCH38 that are either incomplete, so they're gaps, right, or they're computationally simulated. So, for example, the centromeres are actually just estimates of what satellite arrays look like. So it was amazing that 20 years after the human genome, my colleagues Karen Meager and Adam Philippi led the telomere to telomere consortium to complete the first human genome with this truly tour de force special issue of Science with half a dozen papers outlining what a complete human genome really looks like. It adds about 200 megabases of sequence to the genome, as I mentioned. That's about nearly 7% of the primary sequences of GRCH38. And it's pretty amazing in that the sequences that it adds are just incredibly repetitive, right? So the bits of the genome that we have managed to complete are essentially the satellite arrays, the rDNA, the rDNA arrays partially, they're not actually quite complete, and of course all the segmental duplications. These are the sequences that are the very hardest things to assemble in the genome because they occur thousands of times over and all are almost identical to one another. And just by switching your analyses from GRCH38, from the current reference to T directly improves the performance of genomic analyses. Here I'm showing you a plot from the Genome in a Bottle medically relevant genes benchmark, showing improvements in genotyping, switching from 38 to CHM13 for a variety of different sequencing technologies for both SNPs and indels, just showing you the reductions in false negatives and false positives. Right? And this is just simply by switching your reference, nothing else. But of course, any T is an incredible achievement. But obviously, no single genome can represent the genetic diversity of our species. Previous studies have identified tens of megabases of sequence that are polymorphic within the population, that are structurally varying. So as a result, despite the repetitive nature and the copy number polymorphisms that are present, about 2/3 of structural variants have been missed from studies that have used GRCH38. Basically because of reference bias. It's difficult to find structural variants because they are different to the reference. And as a result, even though structural variants affect more bases in our genome than do SNPs or small indels, they've been largely missing from things like association studies up until now, despite our knowledge that they actually generally have individually more effect than any individual SNP or indel. At the same time, there is a general recognition that as a community, whilst genomics is striving ahead and doing incredible things, we are falling behind on diversity. This is illustrated by this plot here, which shows just the number of people who've been included by their ancestry, who've been included in GWAS studies to date. So I'm sure many of you have seen this plot, but it just emphasizes that, as a share of global population, European ancestry is obviously a minority, but it's the large majority of people who've been included in association studies to date. And I think as we move forwards in general as a community, we need to strive towards having a more equitable representation of all people. And so what we've been doing in trying to build a pangenome is to start from the very root of the sort of data structure that we all use as a community to make that a more diverse and inclusive structure. So the Human Pangenome Reference Consortium, this is something that was funded. I'm so pleased that it was funded by NHGRI about 3 years ago. We got started. And the overall aim is to build a better representation of human diversity. And in our first 5 years, we're going to sequence 350 diverse human beings. And in the process, build a comprehensive map of human variation. And because we've now going to deal with not one reference, but 350, we need to come up with new ways of actually computing on that data, right? How to think about how we handle, many gigabases of resolved sequence, and therefore, how do we build new tools to work with something that represents a pangenome? So today, just over the weekend, we put out our initial preprint as part of a number of papers that we've just been submitting that outlines the draft human pangenome reference. And it has been a tremendous amount of work. And if you're interested in any of the details and things that I kind of gloss over today, you can see this preprint. The draft is about 1/7 the size of where we will be in a couple of years' time. But it comprises 47 phased diploid genome assemblies, okay? 94 haploid genomes. And of course, in addition to the genome assemblies, which I'll go through, we have constructed pangenome alignments. So how do you build essentially a representation of all of the homologies between all of those genomes? That's the second key ingredient to the pangenome. And then finally, of course, we need to demonstrate proof of principle. So we've been building tools and applications, and I will show you some of the things and benefits that you can gain from switching. So just to go into the population sampling, I won't say a lot about this today, but initially we recruited genomes from existing banks, and we've used the 1,000 genomes. So 1,000 genomes obviously is a great resource because there are LCL lines for thousands of samples available that are broadly consented so that we can publicly release this data. A key principle here is that all the data that we're making available is publicly accessible to anybody, anywhere, including all the sequencing data, etc. So what we did essentially was go through 1,000 genomes. We use essentially a method of using PCA to attempt to select diversity so that we could attempt to cover as many different subpopulations as we could, with particularly an overrepresentation of samples from Africa, because that's where most of the diversity is. And then, of course, we also selected lines, because these are from cell lines, that had low passage to try and minimize any kind of acquired variation that we might introduce. And all the lines that we've included are from trios. So they're the children of parent-offspring trios, so that we had parental data, which is actually essential right now, or was essential for us to build high-quality assemblies. So this is representation. I think we have samples from 6 continents and a reasonable number of different subpopulations at this point. You'll note that there are some gaps here, and that is being corrected as we add in the additional stuff. So really powering, this is kind of an aside, but really powering where we are now is the huge gains that we've had in de novo assembly over the last several years. So if you haven't been paying attention to the assembly field, you'd be forgiven for thinking that de novo assembly was still difficult and hard and expensive. But things have really changed. So 3rd generation sequencing technologies, in particular the PacBio HiFi, so the circular consensus sequencing, and also the ultra long sequencing from Oxford Nanopore, is really becoming cost effective to allow us to produce incredibly accurate assemblies for reasonable amounts of money. Not cheap, but reasonable amounts of money. And in parallel to those new sequencing technologies, there have been kind of a lot of gains in computational methods. I just dropped down a few different papers that have been influential to me over the last few years. Just really, it is no exaggeration to say these have completely changed the game in terms of de novo assembly. The quality now is night and day better than where we were just a few years ago. And I think one key thing that is worth mentioning is that whereas 2 or 3 years ago, when you constructed an assembly, you generally constructed a clapped representation. That is, there are, most samples are diploid, but the haplotypes would just be smushed together. So you'd get essentially a sort of chimeric representation or a mosaic across the genome. And that's fine as a first approximation, but actually it's going to introduce errors because there are going to be places where those haplotypes are inconsistent with one another. And therefore there's no way of creating a correct assembly. So the only way to get truly correct, telomere to telomere ultimately genomes is to have completely phased genomes. So the genome assemblies we build are no longer 3 gigabases in length, they're 6 gigabases in length because mom and dad are separately assembled. So in order to to create an assembly pipeline for the HPRC, we actually held a bake-off. There's a paper that's in press now that describes that bake-off, where we invited all of the sort of strongest assembly groups on the planet to contribute assemblies of HG002, and then we performed an evaluation and decided. And we gave them essentially a basket of different sequencing technologies to choose from. And after a lot of back and forth, there are a few sort of really good candidates out there. We ended up picking trio HiFiism from Heng Li's group for contig assembly. So the recipe, the ingredients to that are essentially reasonably high coverage PacBio HiFi sequencing data, and then Illumina parental information, just to give us k-mers, essentially high quality k-mers from mom and dad, which allows us to then phase the assembly. So we took that pipeline, did a bunch of development work to kind of clean it up. I won't go into details here, but doing things like adaptive filtering and contamination removal and so forth, and work to ensure that there's a high quality single mitochondrial assembly per assembly. And then we created a refined pipeline that we actually ran on NHGRI's Anvil, so the cloud platform, to create a reproducible pipeline that we could run on all the assemblies quickly and routinely. And we have that now. And if you're interested, you can go see that pipeline for yourself on Dockstore, and lots of people are using it now for assembling different samples. So I'm going to just take you through some of the highlights of the assemblies as a kind of cohort of human diversity, and just impress upon you just how good they are. So the first thing to look at is just the total length of the assemblies. So here I'm showing you, this is just total length on the y-axis of each assembly. And you'll see that for the samples that contain an X chromosome, so these 3 bars, right? We have an assembly size of about 3.04 gigabases on average, okay? That is 99.3% the length of the T2T reference, which also contains an X chromosome and not a Y chromosome in common with these. So we are very close to having a complete assembly of each of these assemblies, not necessarily in just whole chromosome bits, but in terms of total length, we're assembling almost all the sequence. You'll see this box plot is a bit lower, and that's because these are the haploid assemblies that contain a Y chromosome instead of an X chromosome, and so they are correspondingly shorter because the Y chromosomes are a bit smaller than X in general. In terms of contiguity, this is an NGX plot. NGX plot essentially is a way of measuring or visualizing the contiguity of the assemblies, and you can think of the contiguity of the assembly as being proportional to the area under the curve of each one of these lines. Each line here is an assembly. So the HPRC assemblies are here in sort of purplish, and for comparison, I'm showing you GRCH38, just the contigs, so the non-ends, the non-scap, not the gaps, but the contigs of GRCH38. And you can see that the HPRC assemblies are broadly comparable, if slightly less contiguous on average than GRCH38, right? So it's about a 40 megabase contig N50. This midline represents the contig N50. So on average, 50% of our bases are in contigs that are 40 million bases in length or longer. So extremely contiguous. And just for comparison, this is sort of the theoretical maximum that you can do, which is this is the CHM13 assembly, where you get a contig N50 of about 150 megabases. So the upshot of this is, yes, our assemblies are extremely contiguous, but there's some way to go until we match the complete contiguity of GRCH38. And that's essentially resolving of the order of a few hundred gaps. That's what the remaining challenge to get from here to T to T is. It's filling in a few hundred gaps. In terms of base accuracy, we measured this a whole bunch of different ways. Here I'm showing you just the YAC estimates. So YAC is a tool for measuring base accuracy using k-mer libraries from Illumina. And the upshot is that We estimate that there is an error in our assemblies, in our haplotype assemblies, about once every 220,000 bases. Okay, and we measure that multiple different ways, and we're pretty convinced of that number. So that's pretty accurate. We can certainly do better. We didn't actually employ much polishing of the raw PacBio assemblies at this point, and we think that ultimately to get to the reference, we need to be closer to Q70, which is where CHM13 is, which is about 1 in 10 million bases in error. But anyway, still very good. And we actually did analysis of things like frame shifts. This error rate gives us about 10 or about a dozen-ish frame shifts per genome, which are aberrant, for example, in terms of the transcriptome. So those are things that we need to fix. But nevertheless, this is still highly accurate. And if you'd shown me this 2 or 3 years ago, I would not have believed that this was possible. So obviously, an assembly is a hypothesis, right? So other people have said this, right? We have no way of actually directly observing the complete underlying genome. So we have a hypothesis given the reads of what our genome is. And previously, people have proposed that one way, in the absence of a gold standard, one way of evaluating the quality of an assembly is to think of it in terms of a likelihood function. How likely is the assembly given the reads, right? So we essentially developed a method for the HPRC which takes those highly accurate hi-fi reads, which again, they have an error about 1 every 1,000 bases, and they're about 20,000 bases in length each. We take those very accurate hi-fi reads and map them to the diploid assembly. So we're mapping them to both haplotypes, and then we're adjudicating whether or not they belong to the maternal or paternal haplotype very, very carefully, so that we essentially assign each read to its correct haplotype within the genome. And so given that very careful mapping process, this is something done by Grad in my lab in collaboration with Heng, we essentially take those mappings and then essentially use coverage analysis. It's like a Gaussian mixture model thing, fairly straightforward, to estimate how much of the assembly is actually essentially has coverage that we'd expect given the coverage that we have, versus how much is collapsed. That is, there are fewer copies in our assembly than there should be of a given sequence, versus falsely duplicated, so that we've made too many copies, versus just completely unsupported, this thing in red over here. And we basically walk through the genome, assigning to each sort of interval whether or not we think it is correct or whether or not it is an error. And the upshot is, right, so I'm showing you a plot here for all of the different HPRC assemblies. The green is the stuff that's correctly assembled, and then these other colors here are either uncertain or in error. And you'll note that there is a discontinuity here in the green. So this is like up to 70 megabases, but this goes up to like 3,000, right? The upshot is that about 99 or just slightly over 99% on average of our assembled bases are predicted to be structurally correct. We did this with Flagger, but we actually did this with a number of other tools as well to evaluate it, and we're pretty confident in this number. So about 99% structurally correct, with some variation in the amount. And just to assess the false positive rate, we also looked at applying this pipeline to CHM13. And CHM13, yes, we still predict a few bases, about 1/10 of the bases, to be in error. It's about 0.01%. So we're pretty confident that Flagger is giving us an accurate estimation with a small number of false positives of what is incorrect. And in terms of what we see as being incorrect, almost all the errors are in the difficult regions. They're in the satellites, they're in the segmental duplications, they're in the NTRs and STRs. But even despite the fact that there are errors in those sequences, and we know there are errors, when we look at their kind of annotation classes, even beside that, we still see that, say, 95% of alpha satellite sequences are predicted to be reliably assembled. 95%. That is a truly astonishing number, given just how difficult those sequences are to assemble. And similarly for other repeat classes. So one of the things that we can do with our assemblies now is go and look at some of the really, really difficult regions, not in one genome, but actually across lots of genomes at the same time. So it's been known for a very long time that human centromeres vary tremendously in terms of their length. So this is cytogenetics work. Karen sent me the slide, right, from a paper way back when, showing that these are just 2 alleles of, this is chromosome 9, okay, right? So these are 2 just separate alleles. Essentially, this is staining the higher order repeat array of the centromere of chromosome 9. And you can see just tremendous, this is at karyotype level, tremendous differences in the size between those arrays. So then the question becomes, well, what do they actually look like at a base level if we start to characterize them? And whilst it's true that the majority of centromeres in the current draft are not correctly assembled, we identified or flagged an error, there are about 4 of them that appear to be assembled correctly in the majority of individuals. So chromosome 2 centromere, chromosome 9, chromosome 12, and chromosome 8 appear basically to be correctly assembled in the majority of sequences. And we can see, essentially, we recapitulate that previous, these previous cytology marks, showing that actually, yes, we observe tremendous length variation in the centromeres that have been assembled. So just taking, chromosome 2 here, you can see that in one individual of our panel, we have a centromeric length of 2.7 megabases, to 2.7 million bases of this higher order repeat structure. Whereas in another individual, we have a centromere length of just 0.9 megabases. So that's pretty incredible, a more than 3-fold difference in length between those 2 individuals. And that is because we know the centromere is essentially resistant to recombination. It's extremely rare to get recombination within the centromere. And so as a result, centromeres evolve rather like chromosome Y. They evolve essentially, independently along lineages. And so we get this kind of tremendous divergence between individuals, which we can now see at a kind of at a base pair level, which is, just astounding. And I should add that, yes, we validated these predictions both with Flagger, but also with tools from Pavel Petner's lab. And yeah, they all look to be basically structurally correct. So I think that, in the future, as we move forwards, we're going to have, just for the first time, the ability to look at these regions in some detail and see whether or not some of the variations actually associate with any, for example, chromosome segregation disorders. Another interesting, although rather speculative observation involves the acrocentric chromosome short arms. So this was actually rather serendipitous. We started searching for interchromosomal misjoins, so places where our assemblies were joining together 2 different chromosomes, right? And we did this by basically blasting our assemblies against a reference and then looking for places where there was a very high confidence alignment between 2 different chromosomes, 2 different human chromosomes. And long story short, we found one apparent chromosome misjoin that we fixed. It was very clear, and it was a result of an assembly error. But the remaining, about 260 of them, are all between the short arms of the acrocentric chromosomes. So the acrocentric chromosome short arms are essentially repeat arrays of 3 different kinds of repeats, right? So there's the rDNAs, and then there are 2 different types of satellite repeats. They're extremely difficult to assemble correctly. What's interesting, however, is that the most common aberration that we observe cytologically in humans is Robertson's translocations. So these are essentially, they occur about one in 1,000 human beings. Many people are walking around with them, don't know they've got them. Essentially, it's you take the 2 long arms of an acrocentric chromosome and you fuse them together. They often involve, they have often very little impact on the person's life, although they may affect fertility. And it's interesting that we are seeing that these alignments that we're seeing that apparently join together these chromosomes indicate there's really, really strong homology between the different acrocentric chromosome short arms. We don't understand, we wouldn't predict that, given that if we expect them to be evolving separately, they should not maintain really, really high similarity, but they apparently are. And I think that although this could be due, we don't know, this could be due to assembly or alignment error. I don't discount that. It is also possible that what we're seeing here is the result of some kind of interchromosomal gene conversion events that are taking place, or some other mechanism that is maintaining very, very strong homology between these short arms. Something that we completely have been, although people have speculated about in the past, completely had no evidence for. So for the first time, as we build these catalogs, we can start to actually see, what is going on in some of these very, very difficult regions of the genome. Along with the assemblies, we actually created a new ensemble gene mapping pipeline. So this is essentially ensemble, the ensemble browser, recognizing that they have to start to actually be able to annotate not just one genome, no longer one reference genome, but actually lots and lots and lots of reference genomes. And so the upshot is that they take GenCode version 38, and then they very, very carefully map each of the genes and transcripts to each of the assemblies, trying to find its correct location and a valid gene model. And in doing this, they consider the identity and coverage of the mappings, but also the synteny of surrounding genes, and their essentially arrangement and complexity. For comparison, I'm showing you the CHM13 T to T on these swarm plots, showing that our HPLC samples are a little bit worse, perhaps 0.5% worse in terms of annotation accuracy than the T to T, which we would essentially expect to be perfect, but still very, very highly accurate. So more than 99%, I think 99.2% of all protein coding genes are uniquely and unambiguously assignable in each of our assemblies at this point, which is, again, kind of astounding if you've worked in assembly for any length of time. One of the things that we did was go look for gene duplications. So previously, people have used, short read mapping to do copy number variance estimates. This goes back to 1,000 genomes to look at, how copy number polymorphic are genes in the human population. Well, now we can actually look at not just kind of copy number estimates, but actually haplotype resolved, complete loci to see what these duplications look like in sequence detail. And the upshot is that we find about 1,500 of our 20,000 protein coding genes are duplicated in at least one HPLC sample. That's nearly 8% of genes having a copy number variation. Most are rare, so this plot essentially is showing you the number of genomes, the number of samples that contain the duplication, just indexing by frequency, with a small tail of very highly frequent copy number polymorphic genes, okay? When we look at the number of gene duplications that are present per individual, we find about an average of 44 gene duplications. Again, most of them are sort of common, and then there's a long tail of rare gene duplications. It's also interesting that those gene duplications mostly occur outside of known segmental duplications. So these are mostly essentially rare segmental duplications that have never been annotated previously. And when we look at the kind of commonly copy number polymorphic gene duplications, we see a bunch of genes that we would probably expect. So for example, we have ZNF genes, we have GPRIN2, Gage2E. If anybody's familiar with these loci, they're really, really highly repetitive. The gray bars here are just showing you the number of samples that the gene is copy number polymorphic for, and the blue is showing you the actual haplotype counts for each sample. I won't drag you through a lot of dot plots, but we looked at a lot of these different loci to see what they look like, and here's just a nice example. So in GRCH38, this is a dot plot, so I'm showing you the alignment between 38 and just one particular haplotype that we've chosen. You can see that, so this GPRIN2 gene is present once in GRCH38, but we can annotate it confidently at least 4 times in this particular haplotype. That's very common. Most of our haplotypes have this kind of copy number variation. And you can see, but just by the sort of repetitiveness of this dot plot, that the sequence homology is really complex. In general, these gene duplications are happening inside of large, complex tandem duplication clusters where any single reference, the upshot, any single reference is going to be a very poor proxy to the actual haplotype sequence of that particular sample. Just illustrating the importance of having haplotype resolution for when you really want to do fine-grained mapping and understanding of these kinds of loci. So, okay, so that's the assemblies. And now I want to talk a little bit about the alignments, because a pangenome is, yes, it's a set of assemblies, right? Of course, that's, the step 1, if you will. Well, step 1 is recruitment of diverse individuals. Step 2 is assemblies. But step 3 has to be some kind of representation of the common sequence between individuals. And to do that, we've been building pangenome graphs, okay, or pangenome alignments. So first, I want to dispel something at this point, because oftentimes I get this question where people say, well, why do I need to use this complicated pangenome thing, right? It seems really, really complicated. Why can't I just pick one particular reference sample as my representative, right? Because we're all, I'm British, I'll just pick the British reference genome if that was a thing, right? But previous work, and there are multiple reasons why that's a bad idea, but I want to take you through why that just doesn't work very well. So, previous work, and I'm thinking of 1,000 Genomes and HapMap, show that most common variants are global because they're old. They predate all of the divergence of different human populations. Common variant frequencies actually don't vary that much between most reasonable populations. You can use FST estimates, right? They don't vary that much. And the vast majority of alleles, the vast majority of differences between any 2 genomes are common, right? 96 to 99% according to 1,000 Genomes estimates. So the upshot of those observations are, if I take any two randomly selected genomes, I call one my reference and my other my sample that I'm studying, they will have millions of differences between them because most of the differences are common and very old, right? In contrast, a pangenome of just a few hundred individuals will capture the vast majority of common alleles and therefore the vast majority of differences. So the pangenome, just by virtue of representing all of that common polymorphic sequence, allows us to actually, capture the variants that are present in almost all the variants that are present. Not the rare variants, of course. We all have rare variants, right? There are many, many, many rare variants, but the vast majority of alleles are not rare. They're common. So, okay. So the pangenome alignment, I would argue, is the essential data structure for human genomics moving forward. And I want to stress at this point that the use of genome graphs is just a way of representing an alignment. That's all it is. It's just an alignment, albeit a generalized representation of an alignment. Okay, so I'm just going to, for people who many of you will be aware of this, but I just want to kind of go through the way we think about this. Okay, so here I'm showing you an alignment of two sequences, a blue sequence and an orange sequence. And below is the corresponding genome graph. It's a graph, so it has nodes and edges. The nodes there are different ways of formulating this, but the nodes in our formulation represent sequences, okay? So little snippets of DNA. And the edges obviously connect the nodes, and they represent the connectivity of those sequences together. And what's important to understand about genome graphs is that the edges are what we call bidirected. That means that each endpoint of each edge has two endpoints, right? You connect either the left side or the right side of a given node. And that's important because DNA is double-stranded. And so we need to be able to represent strandedness. We need to be able to know whether or not we're going through it forwards or reverse complement, right? So for example, for an inversion, right, it matters if I'm going to go through from right to left, or if I'm going to go from right to right, or 5' 3' or 5' 5', if you will, right? So that's very important. But once you've got that concept, it's just a graph and the edges are bidirected. That's it in terms of the topology. And then the other aspect of a genome graph that's essential for what we do is actually the underlying haplotypes, the way the actual sequences, the assemblies that I've just talked you through, flow through that graph. So here, the blue path represents that blue sequence. So if you traverse that blue path, right, a blue walk, if you will, you'll see that it spells out that blue sequence. Similarly, if you follow this orange path, you'll spell out the orange sequence, right? And you'll see that, for example, there's that inversion there, right? So a genome graph is the graph itself and the sequences embedded within that graph. And that is our representation of the variation and the homology relationships between the sequences, okay? That's it. That's all you have to get your head around. Now, in order to build this for the Pangenome draft, we sort of did a sort of relatively controlled experiment. So we took our genome assemblies. We actually took 44 of the 47. We held 3 out to do various benchmarking exercises. But we took 44, so that's 88 haploid genome assemblies. And then we also included GRCH38 and the CHM13 assembly. And that's important because we actually want to be able to kind of backtrack from the genome graph back into GRCH38 coordinates or into CHM13 coordinates. And by including them as paths in our graph, we know exactly where they correspond to, right? And then, of course, we have an alignment process. I'll get to that in a second. And the output is then a pangenome in GFA format. So it's really important, actually. If you think back to 1,000 Genomes, one of the big contributions from the 1,000 Genomes project was the creation of SAM and BAM and VCF and all those terrible things, right, that we use as a community to work, to get our work done. And the same thing has to happen now as we transition to the pangenome. So we are essentially all kind of congregating or coalescing on for representing the pangenome. We're coalescing on a subset of the GFA format, which represents exactly what I just said. It represents the graph and it represents the paths flowing through it. And it's pretty simple. It's just like SAM. It's a text-based format that is human readable and so forth, and you can see. Of course, as everybody knows, one of the issues with text-based formats, while they're very nice for writing easy parsers and stuff, is that they're not particularly small, right? They tend to be rather large on disk. So we've also created an efficient compressed format. This is work by Yoni Sirin. where we essentially take that GFA, so it's the graph plus the paths, and we store it in a compressed representation called GBZ, which is, if you will, if BAM is to SAM, GBZ is to GFA. And the upshot is that we can take our pangenome, so that's about 270 gigabytes, roughly 270 billion bases of DNA, 270 gigabytes. If you compress that, you get about 70 gigabytes. Now, the GFA for that is smaller because, of course, we've compressed similar sequence by wherever it co-occurs on a node. So the GFA is smaller, but in the GBZ, and if we compress the GFA itself, we can get down to like 11 gigabytes. So, roughly more than a 20-fold compression. But the GBZ, which is accessible and computable without decompression, is only 3 gigabytes in space. So actually the pangenome, even though it represents 100 individuals, because those individuals are all very similar to each other, because we're all very similar, right, is actually pretty reasonable from a computational point of view. So we're really keen on this. There's actually a preprint out maybe today from Yoni describing this. And there are really nice libraries in both Rust and in C++, C, and Python for manipulating this. Okay, so it's at this point that I have to confess that building pangenome alignments is an open question, okay? And it's almost an ill-posed question because it's not necessarily clear how to align together two human genomes. Different people, reasonable people, can disagree because for different purposes, including one set of homologies or not including one set of homologies. For example, do you align together two closely related paralogs or do you leave them unaligned, right? That question is not really very well defined. And so we actually, as a community, came together and devised 3 different approaches for building pangenomes. So first off, Minigraph. So Heng Li previously published this tool. It's really, really nice. He's been updating it for the HPRC. What it is essentially is an iterative approach that starts from the reference and then iteratively adds in each additional genome, just recording the large, so things greater than 50 base pairs, the large structural variants. So you can think of it essentially as a graph that includes the original reference with all the other structural variants kind of bolted on top of it. Okay, it does not include small variants. And so then Minigraph Cactus is Minigraph plus the Cactus genome aligner. So what we do is we take the Minigraph graph and then we essentially refine it with Cactus, which is a whole genome multiple sequence aligner, to add in essentially all the different base level information and refine it accordingly. One thing to note about the Minigraph Cactus is that right now we omit all the centromeric variations. We explicitly cut out the satellites because they are really, really difficult to align. We don't know how to align them correctly yet, so we just omit them, right? So whilst we include the satellites represented in our reference, we don't include the ones in our variant, and that's a limitation right now. And then finally, Eric Garrison's group have developed the Pangenome Graph Builder, or PGGB, and that's a much more ambitious approach, which starts from all of the assemblies equally, constructs all against all alignments, and then induces a combined multiple sequence alignment. And it includes all the centromeric variation. And as a result, it's complete, but it is definitely a lot more complex. Just to give you a sense of this, here's our 3 graphs. Here's the sort of size and cardinality, the number of nodes and edges, I should say, in our graph. And then the total length, which is just the length of the sequences of the nodes in our graph. Okay, and if you look at the Minigraph graph, it's a little bit longer than a human genome. Essentially, it's the human genome plus a bunch of structural variant bases. Once you add in the base level information, you're increasing the complexity of the graph by more than 10-fold, right? It's a lot, more than 100-fold in terms of numbers of edges. But the total length doesn't change that much because we're essentially just refining and adding a lot of detail, but there aren't that many SNPs. And then finally, there's the PGGB graph, and that's much more complex and adds about 5 gigabases of additional satellite sequence. Again, it's really ambitious to try to include this sequence at this stage, and a lot of work is needed to try and figure out how to actually T to T align through these very difficult arrays. So it's somewhat a work in progress. So I'm going to just take you through sort of the basic stats of the pangenomes that we have. So again, so Minigraph is excluded from this because it doesn't contain the small variants. But for the Minigraph Cactus, which we abbreviate to MC, and the PGGB graph, we have about 20-ish million small variants in our graphs. That's about one variant every 100 bases, essentially, of SNPs and indels. And you can see that obviously the vast majority of SNPs are biallelic. That's the stuff that's shaded in dark. But a bunch of the indels are actually have more than three alleles, have three alleles or more than three alleles at a certain frequency. And in terms of numbers of variants, yeah, about 5 to nearly 6 million variants on average per sample per population. This just recapitulates what we know about differences between populations and the amount of heterozygosity. In terms of structural variants, we find about 70,000-ish large structural variants in our graphs. In terms of numbers, that's what I'm showing over here on this left panel. You can see that ALUs are extremely prevalent in the genome, as are VNTRs, as are repeats, as are structural variants that occur outside of repeats. These are essentially sort of non-repetitive structural variants. But in terms of the number of bases that these variants involve, the picture is rather different. So VNTRs contribute a very large number of bases that are structurally variant, as do the seg-dupes. Seg-dupes are much rarer, but they tend to be much, much larger. So they contribute the majority of additional bases to our genome. The other thing to note in this plot is that the most ALUs are biallelic. That's why it's colored dark here. But VNTRs tend to have 3 or 4 or more alleles that are present. And the majority of the large VNTRs are multi-allelic. They contribute a lot of sequence and have a lot of different polymorphism within the population. In terms of numbers of variants, we did this per haplotype, and we find about 18 to 20,000 structural variants per haplotype per assembly. We also were very careful to go and compare all the variation that we created to existing libraries of variation. So we essentially ran standard variant callers and then against the linear reference, and then just compared them. And the upshot is that our variant calls are at least as close to each other as any two not reference-based callers would be in terms of their concordance and agreement. For both small variants, which are the most concordant, but also structural variants. In terms of length, the length distribution of the variants that we see, all across the length spectrum, from very small, so these are insertions going out this way, and these are deletions going out this way, and the frequency of the different variant classes is shown here for the mini graph and PGGB graphs. But in terms of precision and recall, shown here in yellow and blue, we see essentially strong agreement with existing variant sets across the length distribution, suggesting that maybe there's a bit of a dip at the very, very large, but suggesting that the vast majority of our structural variants are in good agreement at all sizes. And you can see encouraging peaks here for example, L1s and ALUs. So the majority of our genome graphs are rather boring to look at. They just look like left to right alignments. But there are, of course, loci that involve copy number variation. And we looked at a bunch of them. Here I'm showing you HLA-A. So this is one of the 3 types of MHC class 1 receptors, I think. Did I get that right? Anyway, HLA-A, this locus has a number of different genes in it that which I'm showing. So this is a bandage plot showing you the graph. This is actually the PGGB graph, but it looks almost the same in the mini-graph cactus graph. And we've essentially colored the genes on this plot. So you can see one here. Here's HLA-H. Here's HLA-A, for example. All right. And interestingly, it's been known for a long time that there is an HLA-Y that's in the population, but nobody knew where it was. It had not been resolved as a haplotype. Well, in our graphs, it is resolved. It is resolved in all of our assemblies. And interestingly, when we trace, this is a detailed plot, but when we trace GRCH38 essentially through our graph, that's this colored path that goes from starts out red and then gets more purple as we go through. You can see that it excludes HLA-Y. So there's a good reason why we don't know where HLA-Y is. It's not in GRCH38, okay? But actually about 28%, nearly 30% of our haplotypes include the HLA-Y insertion. So it's this path that goes this way. There are also other interesting haplotypes that, for example, involve deletions of several of the genes that are present in this locus. So this, we went back and we looked at the literature in terms of correspondence. We see extremely good agreement between our haplotype predictions and the known literature, but we also resolve, things like HLA-Y. There were other examples in the paper correctly and haplotypically for the first time, allowing us to get a better representation of these critical loci. Okay, so in the time left, I want to talk a little bit about applications. So my group previously developed VG in collaboration with Richard Durbin's group, and when Eric Harrison was a grad student and a postdoc with me. And then recently we've developed this VG Giraffe, which is essentially a very fast short-read mapper. It's designed for mapping to a pangenome, and it's about as fast as BWA mem, but able to map to a really complex pangenome. And it's fast because essentially, if all the variation is present in your graph, then and Illumina reads contain almost no indel errors. Almost every read is just a gapless alignment to a path in the graph. And so we just rapidly find essentially linear alignments without ever having to do DP. I won't take you through this. It's published in Science back in December. But we use Giraffe to map to our pangenome. And in this case, the pangenome is like a black box. You just map to the pangenome, and then we just project the alignments from the pangenome, so with all that knowledge, back onto 38. So we're using the pangenome to correctly localize reads, but then we're projecting back into reference space, a standard reference space, to be compatible with existing tools. In this case, we were using deep variant from the Google Health team to then just call variants, okay? And so we did this two ways. We looked at deep variant, which is a single sample caller, and we also did trio-based calling with deep trio, which is just a version of deep variant, which uses parent-child trios for calling. And the upshot is that about 34% of errors are eliminated by just switching to the HPRC pangenome from GRCH38. So comparing sort of standard BWA mem with deep variant to just Giraffe with deep variant, we get about a 34%. This is over 7 different samples from Genome in a bottle, so pretty sure about this number. Reduction in false positives and false negatives. And then switching to trio calling just improves that accuracy yet further. So it's actually interesting that deep variant single sample calling is almost or slightly more accurate in terms of F1 score than BWA mem-based trio calling, right? So essentially the graph beats the trio in terms of overall accuracy. The benefits are not equal across the genome. So when we look at, for example, the challenging medically relevant loci, the gains are much larger. So we're seeing like a percentage gains in accuracy with the switch to the graph. And when we looked at why that's happening, so to try and understand where the improvements were happening, a number of things pop up. But one of them is actually just the ability to recognize gene conversions. So this, I'm sorry, this plot is rather complicated. I didn't have time to sort of summarize it. But here I'm showing you true positives, and false negatives for the Giraffe calls, for BWA mem calls, and these are the Edico Dragon results. Okay, and in red here, these are false negatives. Okay, and you'll see that there are a lot of false negatives, particularly in the Dragon and BWA mem results. So why is that? Well, it turns out that in the reference, there's been a gene conversion event, okay? So part of this gene has been gene converted with another similar sequence. And so reads from that region, from a haplotype that has not been gene converted, just don't map to this region correctly. They map somewhere else. And so you get a whole bunch of false negatives when you do that. But because our graph includes the alternate haplotype without the gene conversion, the reads can suddenly map correctly to that loci. This is not a lone example. We see lots of these examples. Right? This is just a particularly clean one. And when you do that, so we recalled all 1,000 genome samples, right? When we do that, we see that, oh, lo and behold, there's suddenly a cluster of genotype frequencies of about 25%, because that's the frequency of the gene conversion in the population, that are suddenly pop up. So we're suddenly able to resolve this loci just by having knowledge of that haplotype. So it's really actually very intuitive when we start to look at this to actually understand why the graph is really helping us. And it's principally because of structural variation that this is happening. Okay, so the other, probably much bigger win is to look at structural variant typing. So again, I mentioned earlier, structural variant typing has been basically excluded from a lot of our analyses because it's much harder to do than with small variants, right? Because the variants are difficult to find given reference bias. So instead, we used PanGenie to genotype SVs. I'm going to rush through this a little bit. And the upshot is that using the pangenome, we can essentially genotype twice as many structural variants per sample. So we can now genotype the majority of structural variants in any sample using short reads, whereas previously we were at about a third of structural variants being typed by state-of-the-art approaches. Okay, and the benefits of that, the big gains are happening mostly in tandem repeat clusters. So VNTRs suddenly pop out and we're able to type them relatively accurately, and also in low mappability regions. So having kind of basically got knowledge of these variants, we're suddenly able to type them correctly in small variants. And this is going to be really big for GWAS type studies. Yeah. Yeah, I'll rush through this. Lo and behold, the results look highly concordant in terms of Hardy-Weinberg and so forth. We did a whole bunch of filtering to try and establish that everything was good. We also did a bunch of functional assays. We developed an RNA-seq based mapper, and we can demonstrate benefits in RNA-seq mapping in terms of reducing the false discovery rate of mappings whilst maintaining really good true positive rates. So this is a difficult thing to do, actually. We use simulations for this, but we're pretty convinced. So to summarize, okay. So to me, the human pangenome will be vital to ensuring in the future that we as a community, and I talk to everybody in this room when I say that, are equally represented by that core reference structure. It is beholden to us, upon us, to try and make genomics an inclusive and diverse activity for everybody, right? And we need to do that by not just refactoring the reference, but many things. But this is a key step. And the draft pangenome that I presented today adds about 120 megabases of new sequence to the human reference that's copy number polymorphic, about 280 billion bases of haplotype resolved assembly, more than 1,500 gene duplications, more than 20 million small variants, and about 70,000 haplotype resolved structural variants. It's going to take time. So whenever I'm talking to funders and so forth, I stress this, right? No one is under the impression that the pangenome is going to be adopted overnight by everybody, and certainly not, as somebody who works with the UCSC genome browser, half of the hits to the UCSC genome browser are still on GRCH 37, let alone 38, right? It takes a long time to migrate. But if we have patience, and if we have the kind of foresight and ambition, and I think we should be ambitious as a community, to create appropriate methods, then we can move, and we've demonstrated through this initial reference that just by switching in sort of initial, existing analyses like genotyping, that we can substantially improve the state of the art in terms of things like, genotyping. Okay, I'm going to stop there. I had a bunch of future slides, but I will just run through that and thank a lot of people who were involved in producing this. The people in bold are particularly the people whose results I've highlighted. And I just want to stress also that on this paper that I've been talking about today, Ira, Eric, Heng, and Tobias are kind of my partners in crime in terms of bringing all of this together over the last few months. And also to shamelessly plug, if anybody's looking for a postdoc, please do send me an email. Thanks.
So we're going to have questions, and I'm going to take the privilege and ask the first. You may have to repeat it there. As you acknowledge, it takes a long time to change, especially in the clinic. We're still using HE19.
Yep.
And so we're trying to move to 38, and believe me, is very hard, in part because of the annotation, the clinical annotations. And I think that two main questions. The first is, are you guys, or the Human Reference Consortium, are they going to move into the new T2T assembly instead of GR38 along the way to get into the pangenome? Because you show there's already a benefit by using that.
Yep. Yes. So I didn't have time, but I can certainly tell you that the benefits of moving to the pangenome are at least as large as the benefits of moving from 38 to CHM13. Exactly. But when we built the pangenomes for this just draft effort, again, which is just a draft and just really a bunch of experiments, we did include both 38 and CHM13. And by including the existing references, and in the future, we may well be able to include, HE19 in there as well, right? By including legacy assemblies inside of the graph, then we can actually have a path towards backwards compatibility. So that, for example, if you want to run a genotyping black box, well, you run it against the pangenome, but you project then back, your analysis results back onto the clinical, reference of your choice. So I actually think that there is we're still working on this, but there is an opportunity here to make something that's actually backwards compatible rather than just requiring wholesale shift. And yeah, absolutely, we have to include CHM13 just as well as we include 38. And
my last just comment maybe is that, as you say, part of the problem is the clinical annotation, but maybe there's the need to think about not only graph structures and alignments for graph structures, but also what are the structures to store clinical annotation that doesn't depend on a linear reference, which already is failing us, with alternate haplotypes, right? And maybe that's an area that needs some study.
So two things to say there. So first off, I don't believe that aside from some people in this, the kinds of folks in this room, that most people are going to look at the graph ever. What they actually want to be able to do is to get their results on the reference of their choice, which could be just essentially the haplotype that best represents the sample they're studying, okay? At a given location. Of course, that haplotype will vary as you move across the genome. But I think it is totally reasonable to expect in the future for people to be able to essentially use the pangenome to get their annotations projected or translated onto any assembly that is included in the pangenome itself. So future versions of things like the genome browser should allow you to be able to select, for a given gene or a given haplotype, let's say you're looking at MHC, you should be able to choose the exact type of MHC and then see all your results on that version of MHC. That for me is a good future where people, it's understandable. People can essentially interpret things based upon the genetics of the loci that they're looking at. I think that's something that can be achieved, but it's going to take a whole bunch of work from the sorts of people in this room to actually enable that kind of dynamic translation of things like clinical annotations.
Thank you. Let's go with the questions there.
So, in some sense, all the downstream benefits that you showed, I mean, they're really important to show, but also they're expected, right? You would think that was there anything that was kind of shocking that you were like, well, I actually did not expect to see this kind of thing happen?
Well, like whenever you're developing a method or any kind of, that there are always kind of complexities, right? I think one thing that I didn't, this is, I've known this for a while actually, but it was very apparent with the HBRC. Actually, probably not beneficial for a lot of applications to add a ton of rare variation into the pangenome, or at least the current methods that essentially treat all variation as equal don't work very well. In the future, for us to include kind of variation from, say, thousands of individuals, we're going to have methods that really condition carefully on frequency, essentially, or the likelihood that that variation is present in the sample. When you just naively include all the variation, right, then what tends to happen is you actually start to see, performance really dropping off because you end up with lots and lots of spurious mapping. And I didn't think that effect would be very large. Turns out it is a pretty significant effect. And so I think we're actually going to need to be quite a lot smarter about future versions of mapping to think more about haplotypes and more about haplotype structure than we have done previously. So that's, certainly that was something that I didn't anticipate until we really got into this.
And we have only time for 2 quick questions, so please go ahead and try to be succinct.
Yeah, so I was thinking about your talk, in the realm of like cancer biology, where you have like a bunch of cells with very different structures and maybe a lot of structural variants. Could you comment on like how you would use a graph to potentially extrapolate, let's say in your coordinate system, how you would identify like where a variant is, and if, as you were saying, in a form of rare variation, would that be able to be extrapolated from like sort of genome graph?
So I think one of the issues in cancer is that it's actually quite difficult sometimes to discern what is somatic versus what is germline. And partly that's because separating the germline, particularly if you don't have germline normal, is quite a tricky problem. And I do think that Pangenome has a role to play in basically better enabling us to annotate the stuff that we know actually is existing in the population, right? That's actually present in the germline. But the somatic variants, apart from the ones that are really recurrent, and there aren't that many of those, right? Most of them will all be unique and rare and different. I think the hope is that by having a pangenome that represents all the common stuff, it's just easier to see the rare stuff or get the context of the rare stuff correctly. I do think in the future there are interesting methods to develop to think about the population of genomes that are present in an individual in a somatic thing as a pangenome, and to think about those kinds of methods. But that's very different to the reference structures that we're building today, although I hope some of the algorithms will have applicability. But yeah, I think it's an interesting area.
Last quick question.
Great talk. So I think you said that currently we have the complete assembly for 4 cellulars, like chromosome 2, chromosome 9, and coders.
Yep.
So are you referring to the stage and 13 latest version that we 2.0 or 1.1?
That would be 2.0. We use 2.0 in the graphs.